The image you see here on your screens is the ATP generating or payoff phase of glycolysis. Note that in this phase, glyceraldehyde 3-phosphate or GAP is subjected to 5 reactions, which ultimately leads to the production of pyruvate at the end of this phase. In the payoff phase of glycolysis, GAP is oxidized and phosphorylated so that subsequent intermediates of glycolysis can donate phosphate to ADP to generate ATP, thus making up for the 2 ATP molecules that were expended during the preparatory phase. The first reaction in this phase involves the NAD+ dependent oxidation of GAP, particularly its aldehyde group. The enzyme catalyzing this reversible reaction is glyceraldehyde 3-phosphate dehydrogenase, which we'll abbreviate to GAPDH. Recall from previously that dehydrogenases are oxidoreductases that reversibly transfer hydrogen from one substrate to another. In these types of reactions, coenzymes such as NAD+ and FAD act as carriers of reducing equivalents. In this particular reaction, GAP is oxidized to 1,3-bisphosphoglycerate, while NAD+ is reduced to NADH. Now, if you look closely at the chemical reaction in the previous slide, you'll see that this step involves phosphorylation coupled to oxidation. On further scrutiny of this chemical reaction, you'll see that there is actually an endergonic reaction coupled to an exergonic reaction. The oxidation of GAP, the first reaction, is an exergonic reaction that powers a second endergonic reaction, which is the addition of inorganic phosphate to GAP. Because of this, the resultant molecule, 1,3-bisphosphoglycerate, which is in the oxidized state, now has a high-energy acyl phosphate. This molecule is subsequently utilized in the next step of glycolysis, which is overall exergonic. And that next step is shown here in this slide. The enzyme catalyzing this step is phosphoglycerate kinase. The energy released from the hydrolysis of 1,3-bisphosphoglycerate facilitates the transfer of inorganic phosphate from ADP to form ATP. This step in the payoff phase is basically where the first ATP molecule is produced. Since there are 2 GAP molecules for every glucose molecule, there are essentially 2 ATP molecules produced. Technically, the ATP expenditure of 2 molecules in the previous phase has already been paid off, although there is still another ATP forming step in the glycolysis pathway, which we'll encounter soon in this video. To repeat what I just said, the energy from breaking the acyl bond of 1,3-bisphosphoglycerate is high enough for transfer of inorganic phosphate to ADP to be energetically favorable. Also, the initial investment of 2 molecules of ATP in the first phase is recovered in this step. This manner of ATP formation is still substrate-level phosphorylation. Additionally, this is the only reaction of glycolysis that involves ATP, and yet is reversible under normal cell conditions. Normally, we would think of ATP formation as a unidirectional process. The product in this step, 3-phosphoglycerate, is left with 1 phosphate group engaged in a phosphoester bond. This bond, however, is of low energy. This then begs the question, can this low-energy phosphoester bond be converted to a high-energy phosphate bond? The answer to that question is yes, through a 2-step process, the first of which involves moving the phosphate of 3-phosphoglycerate from the carbon 3 position to the carbon 2 position. And that particular reaction is actually the next step in the glycolytic pathway. It is a reversible reaction that is catalyzed by phosphoglycerate mutase. Mutases, by the way, belong to the isomerase class of enzymes, and they are responsible for shifting a functional group from one position to another within the same molecule. The second of this 2-step process involves a reversible dehydration reaction catalyzed by an enzyme called enolase, which is also known as phosphopyruvate hydratase. The product of this step is phosphoenolpyruvate, which can be abbreviated as PEP. This step is actually the ninth and penultimate step of glycolysis. The enol phosphate bond on PEP is a high energy bond. And that is basically how the low energy phosphoester on 3-phosphoglycerate is converted to a high energy one. The final step in the glycolytic pathway involves hydrolysis of phosphoenolpyruvate or PEP to pyruvate with concomitant formation of ATP. It is in this step that the second ATP molecule is generated. Again, since there are two gap molecules that enter the payoff phase for every molecule of glucose, there are two ATP molecules produced in this step for every glucose molecule. Now, this reaction, catalyzed by pyruvate kinase, is a highly exergonic one. The high amount of energy released from hydrolysis of the enol phosphate bond of PEP allows the transfer of the phosphate group from PEP onto ADP to form ATP. Overall, the reaction is unidirectional from left to right. Additionally, the loss of phosphate from PEP yields an enol form of pyruvate that spontaneously tautomerizes into the ketone form. That tautomerization is featured here in this image. Note the highly negative delta G naught prime of the hydrolysis of the enol phosphate bond of PEP. This tautomerization is actually important because it effectively lowers the concentration of the reaction product in this last step of glycolysis and drives the reaction towards ATP formation, on top of the overall delta G naught prime of this step being highly negative. Now that concludes the glycolytic pathway. We can actually reduce these 10 steps into an overall net reaction wherein there is a net production of 2 pyruvate molecules, 2 NADH molecules, 4 protons, 2 ATP molecules, and 2 water molecules for every glucose molecule that enters the glycolytic pathway.